Hello, everyone. My name is Jillian Kemsley, and I am an Associate Editor with CNEN and moderator of today's webinar. Thank you for joining today's webcast. Through CNEN webinars, we hope to deliver critical information to select audiences in a timely and interactive manner. Although the format may vary, all webinars share a few things in common. First, each webinar comes to you through funding by a sponsor. CNEN works with sponsors to identify topics of interest and value to CNEN's audience and that are consistent with CNEN's mission. In all cases, a webinar topic would be one that CNEN would cover or is already covering in its news gathering operations. Second, each webinar will be archived at CNN online and at the sponsor's website after the live webcast. This way, those who could not join today can access the information and discussion at a later time. During the webinar, you can adjust the size of the slides on your screen by grabbing the lower right corner with your mouse. If you need technical assistance, please look at the Help widget at the bottom of the screen or type your query into the Q&A box. If you are disconnected during the webcast, please log in again according to the instructions you received earlier. Each webinar's success depends on your participation. Please feel free to ask questions at any time during the presentation through the Q&A box on your screen. The questions will be answered at the end of the presentation, and as your moderator, I will be posing as many as time permits. Lastly, CNEN does not endorse any company, products, or services that may be mentioned in the webinars. Having said that, I'm delighted to welcome you to today's webinar, which is being sponsored by SCIEX. SCIEX is based in Massachusetts and specializes in mass spectrometry instrument systems and software for analysis in basic research, drug discovery and development, food and environmental testing, forensics, and clinical research. Presenting today are Dr. Mauro Aiello and Dr. Dick Kibbe. Dr. Aiello is a Senior Product Manager for Pharmaceutical Quantitation at SCIEX, where he works on developing new technologies for improving quantitation in LC-MS. Prior to joining SCIEX, Dr. Aiello worked for Environment Canada to develop analytical systems to deploy in the Canadian Arctic, and then he received his PhD from Canada's York University. Dr. Dick Kibbe is an Assistant Professor of Endocrinology and of Cellular and Molecular Physiology at the Yale School of Medicine. Dr. Kibbe went to medical school at the University of Texas Southwestern Medical School, where he also earned a PhD in molecular biophysics. He then moved to Yale in 2000 to do an internship and residency in internal medicine, followed by a fellowship in endocrinology. His current research program focuses on understanding the origin and development of diabetes mellitus. I will now hand the presentation over to Dr. Aiello to get us started.
Thanks for the introduction, Jillian. Today I'll be speaking about a product we launched at the last ASMS conference called the SCIEX Selexion technology for 5500 systems. I'll be discussing some of the theory behind the operation of this technology, as well as a brief look at the type of application where this technology can be used. I'll then hand over to Dr. Kibbe, who will go into more detail about some of the applications where he's been using Selexion. Here's a look at the 5500 system with a Selexion system installed on it. On the top of the instrument, you can see the power supply for the Selexion system, and immediately behind the Turbo V ion source, you can see the actual mobility cell installed. You'll also notice a solvent bottle connected to the power supply on the top of the system. There's actually a small liquid pump built into the power supply, and I'll discuss the purpose for this more later on in the presentation. The purpose of this differential mobility device is to separate isobaric species before entering the mass spec, which can be used to do various things such as eliminate interferences in an assay, reduce background noise, and also potentially simplify sample prep techniques and shorten chromatography. This is a closer look at the system installed on the front of the 5500. On the left we have the orifice in front of the 5500. In the middle picture we have the mobility cell itself, in the center of which is the actual mobility region made up of two flat steel plates. As well, on the outside you can see an extension ring which allows the Turbo V source to couple to the front of the system. Finally, on the right we have the curtain plate installed in the system to keep everything clean and prevent contamination from entering the mass spec. You'll notice that all of the components can be installed using thumb screws, so that the user does not need to use any tools to mount or dismount the device on the front of the mass spec. Also, since the Selexion mobility cell is mounted in front of the orifice, there's no need to break vacuum. Because of these design features, Selexion cell can be mounted or dismounted from the front of the 5500 in a few minutes without breaking vacuum and without the use of tools. As I said previously, Selexion mobility cell is made up of two flat plates that are shown at the top of this figure. Differential mobility spectrometry, or DMS, is a term used for systems with this planar geometry. Ions generated in the source on the left side of this figure are pulled towards the mass spec using a gas flow. Ions are separated within the mobility region by applying what's called a separation voltage, or SV. The waveform is an asymmetrical RF voltage applied to the plates. When we apply the SV voltage, ions entering the cell will in general have a trajectory going to one plate or the other based on the SV alone. We can steer the ion of interest toward the orifice by applying what's called a compensation voltage. That ion will then be directed towards the orifice and will enter the mass spec, while other ions are removed. We can then select which ion enters the mass spec by simply tuning the compensation voltage for the ion of interest. This gives us a way to separate ions based on something other than mass to charge before entering the mass spec, and so it gives us the ability to improve selectivity in cases where mass to charge alone is not enough to give adequate selectivity. Some of the advantages of the planar geometry used in DMS are short residence times and fast voltage changes to give us short cycle times of about 25 milliseconds. So roughly 20 millisecond pause time and a 5 millisecond dwell time can be used for quantitative assay. This is, of course, important in cases where fast chromatography is being used, and with narrow chromatographic peak widths, we need short dwell times to get enough points per peak. It also allows what's referred to as transparent mode, where by simply setting the voltages to 0, all ions can be transmitted into the mass spec. It has minimal diffusion losses to maximize signal, and also has homogeneous electric fields and uniform conditions, both of which allow the addition of chemical modifiers, which I'll discuss a bit more in the next few slides. Also, not related to the geometry, but important to note, is the system also supports what's called scheduled MRM for assays that are looking at many analytes in the same analysis. Okay. As I mentioned earlier, the system has built in a small liquid pump that allows us to infuse into the system what's referred to as a modifier. The liquid's infused into the curtain gas where it evaporates and fills the DMS region. Ions entering the region can then interact with the modifier. So why do we do this? On the top, we have a mixture of 11 analytes infused into the system with only nitrogen used as a transport gas. As you can see, it's difficult to separate these ions using mobility alone. On the bottom, we have the same mixture infused into the same DMS cell, but this time using a chemical modifier. As you can see, this gives a dramatic improvement in the separation power of the system. The modifier used is not complex. It's simply 1.5% isopropanol. In fact, IPA is used most commonly for this purpose and works quite well in most cases. But the analyst is free to use other modifiers if they want to try something different on a difficult situation. This basically gives the user some options to deal with their difficult analytical challenges. Okay, so what's happening in the Selexion cell when we introduce a modifier? Ions in the DMS interact with modifier molecules and go through a clustering and declustering process as they go through the low field and high field in the DMS cell. So if we think about the mechanism of separation in a mobility device, it's based on the size and shape of the ion and the difference in its mobility in the low field and the high field. So the bigger difference we can create in the size and shape of the molecule, the bigger the separation power of the device, and more likely we are to be able to separate it from other ions. Next, I'd like to show a quick example of the kind of application where we can take advantage of the Selexion technology. In the interest of time, I won't go through too many applications today, but rather I'll let Dr. Kibbe take us through some of his work with Selexion. The example I'll talk about today is quantitation of clenbuterol in urine samples. This assay is known to have large interferences and also highly variable interferences from subject to subject for all major MRM transitions. Here we'll look at the use of Selexion for the purpose of increasing selectivity for this method. On the top we have the standard Q-trap 5500 for clenbuterol in urine. As you can see, all the major MRM transitions have very poor selectivity and are not reliable in terms of quantitation. Also, it would be difficult to try to decide on the peak integration and exactly how to integrate any of the MRM transitions at the retention time of the analyte. On the bottom, we have the same sample run using Selexion technology on the 5500. As you can see, the 3 MRM transitions are all much cleaner and it's much easier to quantify the analyte with greater reliability. The analyst would also have a much easier time deciding on the integration of these peaks. With the higher selectivity that Selexion gives you, you can now start to think about simplifying complex sample prep methods if you're using them to get rid of interferences, and also with cleaner chromatograms you can start to think about speeding up your chromatography. In terms of robustness, we examine technology in many different ways during development to make sure it's robust. This is one example of the kind of experiment that we do to look at robustness. Here we have 1,000 plasma samples injected over 66 hours for the analytes buprenorphine and clenbuterol. On the top you can see the raw peak areas for these analytes over those injections with a reproducibility of better than 7% and also no real drop in signal intensity over the course of the batch. On the bottom we have the IS-corrected data with reproducibility at better than 2%, so it's very usable in a regulated environment. It's a brief summary of some of the topics that I've covered today. First, we discussed the use of Selexion as a way to improve selectivity. It's available either on a new 5500 system or also as an upgrade to either triple quad or Q-trap 5500 system. It's robust and reliable for use in a regulated environment. It could potentially help improve throughput in cases where you're doing extensive sample prep or long chromatography. And finally, if we look at the Q-trap 5500 with Selexion technology, some really powerful tools for the analyst to develop methods. You have very high sensitivity in MRM mode. Using the Q-trap, you also have very high selectivity for analytes that have the possibility of being fragmented twice using MRM cubed or MS cubed for quantitation. And now with the Selexion system, you also have the possibility to combine DMS with either single MS for analytes that don't fragment well, or DMS with MRM to really improve selectivity. So together, these sensitivity and selectivity tools really give the analyst a lot of options when they're trying to deal with difficult analytical situations. Thank you for your attention. And at this point, I'd like to hand over to Dr. Kibbe.
Thank you, Mauro. Happy to be able to share with you some of the work that we've been doing in my laboratory looking at metabolic flux and why we turn to DMS type of separations for those purposes. So the title of my talk is Opening Doors to Glycolytic and Mitochondrial Flux Measurements and using mass labeling where it meets differential mobility. So just an overview of what I'm going to do is describe how we're using mass spec as a tool for measuring metabolic flux, identify some significant barriers to these measurements, in particular those of isobaric species, and discuss the use of differential mobility separations as a tool for distinguishing relevant isobars, and apply DMS to in vivo metabolic flux analysis. And this is all brand new information. We've had the Selexion now for about 4 or 5 months, and sharing you some of our development with that. So the problem that I'm interested in is that of beta cell metabolism, trying to understand how beta cells work. And the conventional notion is that glucose is metabolized in the beta cell by the mitochondria to make ATP, which goes up and closes the KATP channel, depolarizes the cell, calcium rushes in, and insulin goes floating out. And this is an important, tightly regulated process, because if you were to release all the insulin in your pancreas at once, that would lead to death. And while this notion populates most of the textbooks that are available, there are certain reasons to believe that it's much more complicated than the simple picture. And in particular, if you knock out the KTP channel, you can still have the same process going on. So a number of labs have begun to look at alternative metabolic pathways that can explain what's going on in these beta cells. And I'm showing some highlights of some interesting metabolic fluxes. And it's also interesting to note that, if a cell was designed just to make ATP, it would not have a lot of these metabolic fluxes present. And so our goal has been trying to sort out which of these are most relevant. One of the key fluxes, which is highlighted in this yellow arrow at the end, is pyruvate carboxylase, or PC flux. And this turns out to be a rather important flux that would detract from the synthesis of ATP and could contribute to some other metabolic signal. So one of the things we had done initially was just to do a broad survey of every metabolite from glucose to creatine phosphate, which I have shown here. And we've been trying to be as thorough as possible, connecting every sequential metabolite in the metabolic pathway. And these cells respond to the addition of glucose by changing their intracellular metabolites. And so that's what's depicted here. Going into the depth of the screen, you see the change in concentration of each metabolite with respect to each other. Now, this is a useful picture, but from this it's hard to tell what's going on, and frankly, concentration really only tells you part of the problem. So let me give an example of how concentration may not represent everything. So every summer, my family and I like to drive down from Connecticut to North Carolina to hang out on the beach, and we're routinely passed with a couple loops of interstate traffic on the way down. The first is up towards the Baltimore area, and the second is around D.C., and we try to navigate our way through these. Now, knowing the number of cars on the roads doesn't give us much information as to what the best route to take is as we're going south. But knowing where flux has changed, where maybe there's traffic, as you can see over on the left side of the screen, there's a block on the west loop of D.C. Knowing that makes us know that we need to take another route and go through the eastern loop. And so knowing how traffic is moving through metabolic pathways is very similar. And not just knowing the concentration is important, because some things may not change their concentration but have significant metabolic flux. And to me, the traffic patterns in the Baltimore D.C. corridor look very similar to the metabolic pathways in the cell from glucose down to phosphocreatine. And so what we really want is a method to be able to measure these well. And towards this, position-specific 13C mass isotope labeling offers a number of strategies for these metabolic flux measurements. And to give you some background of how we've used this before in our beta cell, knowing that anaplerosis is important for insulin secretion, which was that yellow arrow I showed you earlier and is depicted in this slide as the PC arrow, we knew that was important. And we had also discovered that mitochondrial GTP synthesis was important. And In beta cells, we made the hypothesis that an enzyme within the mitochondria, mitochondrial PEP-CK, can couple the metabolism of GTP made by the TCA cycle with oxaloacetate to make PEP. And that PEP can then escape into the cytosol of the beta cell to act as a signal for insulin secretion. And previously, methods had demonstrated that there was a lot of cycling of carbon in and out of the mitochondria, and that was believed to play an important role in generating this signal. And what was not clear was what pathway that was going through, and that was in part because of the types of techniques that were available at the time. So our approach to sorting this out was to, one of the competing pathways was this pyruvate cycling pathway compared to the PEP cycling pathway. And our way to sort this out was to use mass spec and use a 13C labeled pyruvate. Pyruvate comes in distal to glycolysis. And since PEP, or phospholipopyruvate, is made both in glycolysis and from this mitochondrial pathway, we needed a way to distinguish one from the other. By putting the label in the third position of the pyruvate, we could track its metabolism in the beta cell mitochondria either through the TCA cycle or through pyruvate carboxylase, where it will label oxaloacetate and malate, which are in equilibrium with each other. If they go through this mitochondrial PEP-CK pathway, they'll generate position-specific labeled PEP, which will come and mix with the whole cellular pool of PEP. And by knowing the enrichment of the precursor OAA malate pool and the product pool of PEP, through very simple calculations, we can determine the relative rate of mitochondrial PEP production relative to glycolytic PEP production. And when we did that, we were quite surprised to see how significant the flux was. Not only did it go up to almost 40% of the rate of glycolysis, but it also went up increasingly with higher glucose concentrations, suggesting that this was a high-throughput glucose-driven event, and suggested that it might be very important for insulin secretion. So that's one example of where we can look at a single metabolic reaction and use mass spec to isolate the different isotopologues and get real flux information. But what we're really interested at the end of the day is getting much more complicated metabolic flux measurements, and using 13C labeling approach to do this. Now, this field really was driven by the folks in doing NMR isotopomeric analysis, which has a wonderful sensitivity for knowing position enrichment. And again, the idea is you track position-specific carbon fluxes through multiple metabolic pathways, and taking advantage of the NMR resolution, you can build estimates of metabolic flux simultaneously through multiple different pathways by modeling the flux and the potential labeling patterns. Most commonly, glutamate is used for these studies because it's a concentrated metabolite in the pool, and it's in interaction with multiple levels of metabolism within the cell. And just to go through this, shown at the top left-hand corner are labeled and unlabeled pyruvate coming from uniformly labeled glucose. The labeled carbons are in light blue, and the unlabeled are in yellow. As they enter into the mitochondria, the carbons from pyruvate go specifically into the 4,5 position of alpha-ketoglutarate, which is shown below. And so labeled or unlabeled will give a specific pattern if it goes through that pathway. Also, as I mentioned, there's pyruvate carboxylate flux in these, and pyruvate will also specifically label the oxaloacetate in those pools. And those oxaloacetate carbons will go into the 1, 2, 3 position of glutamate as the TCA cycle is turned. And that will lead to this group of labeled oxaloacetate species as the TCA cycle turns. And then as we get a second turn of the TCA cycle flux, again we have pyruvate labeling the 4, 5 position, and TCA cycle and PC flux labeling the 1, 2, 3 positions. And if we keep turning the cycle, we get more and more complicated spectra of labeling. But it turns out that once you reach metabolic steady state, which is about 4 hours in these cells, then the patterns even out, and you can use your modeling to determine what's going on. Now this is using one point to get at a lot of different fluxes, and it uses a lot of assumptions. And what we're hoping to do using mass spec was to give more direct measurements of metabolic flux. And again, highlighted in the squares are the molecules that are being studied by NMR. So we're using mass isotopolog multi-ordinate spectral analysis. And in some cases, mass spec with good fragmentation patterns can give us some position-specific information. We're taking advantage of that, as well as the ability to measure multiple metabolites within a metabolic pathway to get some sense of overall metabolism. And we can also look at this under non-steady state conditions, where the spectra are much more simple and easy to interpret, and we can get a direct readout of what's going on. And so here, if you look at pyruvate carboxylate's flux from pyruvate and PDH flux, it's labeling the same patterns, but now the end product is very different. And as it turns out, we can measure both the oxaloacetate and the glutamate by mass spec, and we can fragment the glutamate so that we're only looking at the 4, 5 or the 1, 2, 3 carbons. And now we have a much simpler way of looking at this. So can we do this in real cells? And the answer is yes, we can. We put uniformly label glucose into the media and look at the uptake of the glutamate into the 4 or 5 carbons of glutamate. And when we do that, and we're quenching the cells at different points of time, we see a very nice linear buildup of the labeled carbon into the 4 or 5 position of glutamate, and that's a direct indication of PDH flux. And not only can we look at the overall labeling, we can tell if there's 1 or 2 carbons that are labeled, and it's not until we get out to 30 minutes that we're starting to see addition of a single carbon into the acetyl-CoA pool, and that would be indicative of pyruvate cycling, suggesting that there's very little of it going on very early on in metabolism, which is important for insulin secretion. So we can also look at citrate synthase flux, which is the next step in the path. So acetyl-CoA from pyruvate dehydrogenase will combine with oxaloacetate to make citrate as the first reaction in the TCA cycle. And we can measure and see whether the carbon or the rates of the carbon showing up in each half of the glutamate molecule. And we see virtually 100% of the glucose that's going into glutamate into the PDH portion is being labeled, whereas we're getting a slower turnover of carbon coming into the 1, 2, 3 positions of glutamate from the TCA cycle. And that's giving us information about how long it takes carbons to move through the TCA cycle. And basically what we're seeing is about 10 to 15 minutes of delay is what's required to move those carbons in to those pathways. And we can also look at the labeling pattern of malate, which goes directly into oxaloacetate, and determine whether or not those carbons are coming from the pyruvate carboxylate pathway or the TCA cycle pathway. And we're seeing two different rates of labeling for each of these. And it turns out that all the TCA cycle carbon is what's actually going into the glutamate, and not the carbons coming from PC based on their labeling pattern and their enrichment pattern. And so we're able to track the different segments of the TCA cycle flux from pyruvate into acetyl-CoA as it comes into glutamate, and then as it goes back into the TCA cycle, turns around and makes another cycle through citrate synthase. And we're seeing kind of a surprising phenomenon that while flux into PDH is rapid, there's again about a 10 to 15 minute delay before that labeled carbon in alpha-ketoglutarate makes it all the way back around the TCA cycle to label malate. And then once it gets to malate, it moves on very rapidly to the TCA cycle. And we could also measure the relative pyruvate dehydrogenase and pyruvate carboxylase fluxes and see that both are robust. So the model that I showed you earlier now appears to be a little bit of an oversimplification by looking at multiple metabolites within a metabolic flux. Now we can really start to tease out some intricacies of metabolism, and what we're seeing is that the pyruvate carboxylase pool of oxaloacetate, even though it is made within the matrix of the mitochondria, is distinct and separate from the TCA cycle flux in the beta cell, and that insight would not have been present if we had just used the standard approaches of single metabolite analysis. So we want to do better than this and get a much better sense of how metabolism is working in these cells, and that's where we run into the isobar problem. And it turns out that intermediary metabolism is absolutely full of isobars, and MRMs are helpful if you have a unique daughter ion, and exact mass is helpful if actually if they're not isomers, then you can sort them out. Liquid chromatography is good in the event that they have separable characteristics. And then you also have to take into consideration that there is some chemistry going on during ionization that will interconvert some of these. And so what you really need is a method that can adequately get the metabolites separated from one another without overlap. Now, when you throw mass labeling into this, then that greatly increases the number of potential isobars. So let's say we have two similar compounds that are normally 2 Daltons apart. If they have 3 carbons in them and we put in a labeled carbon source, then all of a sudden we can start to have overlap of the carbons from below with the ones from above, or vice versa, just by the addition of the mass label. So that greatly complicates the spectrum, and where you wouldn't have overlap before, now you do have overlap. And this is particularly sensitive to really contamination from other signals, especially when we're working in vivo. In vivo, we can't get really high enrichment levels just because of expense and practical constraints. And so we really need to know very closely the enrichments down to 1 to 2%. And just as an example of this, I'll show you glycolysis, which is an isobaric nightmare. So highlighted by these different squares are true isomers, where you have the position of a phosphate that's different. The chemical structure is the same for each of these compounds. then compound that with the presence of other isobars on off-pathway metabolism, and then you start getting into problems. And these, glycolysis does not exist by itself in the cell. Many of the metabolites feed into other really important metabolic pathways, such as the pentose pathway and glycogen synthesis, to name a few. And so we can't ignore contributions going into or coming from these pathways. We really need to measure them directly. And here listed are a lot of our compounds within the glycolysis or in the off-glycolysis pathway. And shown in the orange asterisks is that each of these, with the exception of pyruvate and lactate, have a very common daughter ion, which is phosphate. This is the predominant daughter that we get from all of these, so we can't use that to distinguish one from the other. The last two on the list, pyruvate and lactate, do not fragment very easily, and so we're left looking at the parent ion for most of these. And then shown also are some of the overlaps. So in the red brackets we have the true isomers where we can't separate from them one from the other, and especially look at the hexose phosphates where we have 4 true isomers that are metabolically relevant and present in the cell that we can no longer distinguish from each other. And then shown with the green brackets are areas where we could have potential isobars from the incorporation of 13C as a mass label, where now all of a sudden we're starting to get intersection of these metabolites where there wasn't any before. And then a lot of these will lose a water or form a double bond, lose 2 hydrogens, and create, during ionization, and so it really becomes a chromatographic challenge to separate all of these from each other. And what we really want to do is be able to separate things that may chemically interconvert from each other in the best case scenario and let the mass spec do most of the work. And so our goal is to develop a high throughput method for quantitative measurement of glycolysis and gluconeogenesis that's compatible with 13C mass labeling studies, but really is compatible for global metabolomics in general. And that's where we turn to our friends at SCIEX when we heard about their differential mobility spectrometry. And what's really nice is it has continuous separation that's orthogonal to the LC and to the mass spec, so we can run things as we were before, but just put this in front for the pre-ion filtration to add an additional level of selectivity. And this has also led to improved ion transfer in some cases. That's not quite explained. There may be some focusing of ions. Since this is after ionization, it may have to do with declustering of adducts or other things. But we're also getting rid of a lot of background, and that has also played a very important role for many of our species, especially if we're looking at parent-to-parent transitions. There can be a tremendous amount of background that interferes with our signal. And we've really seen remarkable selectivity for being able to distinguish isobars, and I'll show you a little bit of that data in just a few minutes. And our hope is that it's going to redefine the role of chromatography, as in part to reduce matrix suppression and also to give nice peak shapes that are spread out so we can get multiple points across them, but also use it to focus on separating metabolites that are susceptible to interconversion through ionization chemistry without having to move to really, really long chromatographic runs, and this will increase our throughput. So this is not beautiful chromatography, but it's a mixture of all our samples, and we're getting some separation over about a 4 to 6 minute run. and we thought we'd use this as kind of a starting method to see what we could do to get separation for each of our compounds. Now, as Mauro went through earlier, there are a number of different variables that can be controlled in the DMS, and it turns out there's quite a few. And so the number of combinations of these was daunting to us, and so we had to find some way to sort things out to make things simpler. Now, the first 4 of these combinations have to be consistent to all metabolites through the run. You can't change these during the middle of a run. And so we thought we would first set on the general front-end conditions for this before looking at the separation voltage and the compensation voltage. So our first approach was just to look at different modifiers on different combinations of metabolites that we had. And here is a mixture of citrate and isocitrate, which are isomers. The hydroxyl group has just moved 1 carbon from each other. And here we're using 4 different chemical modifiers in the gas phase during the separation. And what we see as we're going from the right to the left within each of the panels is we're increasing the separation voltage. And as we're doing that, we're getting an increase in the compensation voltage that's needed to keep the ion on track into the mass spectrometer. And we see very different patterns depending on the chemical modifier used. In this particular case, isopropanol gave the biggest amount of spread, and acetonitrile was next, and acetone, and then followed by methanol. But we saw a different pattern when we looked at different combinations of isomers. These are the hexose phosphates that we're interested in separating out. And here we see that acetonitrile was giving us the best separation. And again, a different pattern when we looked at DHAP and glyceraldehyde 3-phosphate, again 2 isobars, and a lot of different behaviors. So we figured that it'd be better to look at everything all at once, and so we combined all our standards that we had together, and we infused them, and we did a matrix of the COV at a particular separation voltage over the whole mass to charge area. So we're just looking at all the parents for each one of these. And you can see we're getting different separation patterns if we're using methanol or acetone or isopropanol, but what we did see is that the isopropanol led to the greatest separation of all the metabolites, and so we thought that was going to be the best solution for our particular standards of interest. And then when we infused some lysate using the isopropanol as a modifier, again we were able to see pretty good separation of the metabolites that were in that sample. So we had to also settle on a modifier flow rate and temperature, and this is just giving some example of how we can separate things out more or less depending on the flow rate and the temperature. And here are 3 isobars or potential isobars, 2PG, 3PG, and phosphoserine that we're able to separate. The y-axis is the intensity, and the x-axis is the COV, or the compensation voltage, for a fixed separation voltage. And ultimately we ended up moving with a low modifier and a low temperature because that seemed to work best with most of our samples. And so then we looked at differential mobility separation of isobaric metabolites, and using pairs of or optimized separation voltage and compensation voltages to achieve the best separation. Again, here we, as shown before, we see the effective COV range for a number of different SVs for the citrate-isocitrate mix. And as you note, as the SV is getting higher and we're moving more from the red to the blue side of the scale, we're losing some intensity. We reach a minimum, and then actually we start to see the intensity creeping up. And we've seen this and other different patterns for each of the different metabolites that we've looked at, depending on the separation voltage. And just to give you a sense of how separation voltage affects this, again, we've got our complex sample with a bunch of different masses, and we're going to ramp different separation voltages and look by scanning out the compensation voltage to see where things are. So at a low SV of 1000, we see very little separation along the x-axis. But as we start to step the SV up higher and higher in increments of 500, we see further and further separation of all these isobars to where we're really getting the best separation for each of them. And as I mentioned before with the isocitrate, we're seeing changes in intensity and even some broadening. So this particular mixture was of 2-phosphoglycerate and 3-phosphoglycerate, or 2PG and 3PG, at the same time. Shown above is just the intensity versus COV mapping, whereas below is the heat map of the SV versus the COV scanned out for this combination together. If we start from the right where the SV is low and look at the blue peak in the top panel, that's basically no separation by the DMS cell, and that's what our starting material is from both. As we move further up the SV scale, you notice along the heat map it starts to diminish and fade out in the pink area. The pink peak shown above, we see a reduction in intensity. And interestingly, if you look to the right of that pink peak, you see two smaller pink peaks, which are metabolites that we're separating out from each other. And these can be visualized in the heat map below as the orange to yellow flare that's coming out. And all the green that's in the panel are non-specific noise that we're getting rid of as well. Now, as we move to the higher SVs, we're able to get clean and complete separation of 3PG from 2PG, and we do see a little bit of peak broadening from this. But what's interesting is those two peaks were before, so if you combine those two peaks together, we're about 2 to 3 times the intensity of where we were before getting any separation. So again, we're having some additional effect on either declustering or ion transmission into the MS that is beneficial. So this is the bottom half of glycolysis, heat maps from pyruvate, PEP, 2PG and 3PG, and what you can see is the curves all have different shape, which is nice, which means that we can separate them from each other by the different SV-COV combinations. And if we overlay them, it becomes more apparent that we're really able to get very nice separations of these compounds, which were all infused at the same time into the mass spectrometer. So no column separation and terrific peak separation. Here's another example of being able to separate the 2PG, 3PG, and phosphoserine almost completely from each other. Interestingly, when we ran these purified samples, we see a little bit of pink under the blue and a little bit of blue under the pink, despite the high separation, which suggests that actually there was some contamination of the other species within each of our standards. Now, can DMS resolve all isobars? And I'll bring you back to the case of the hexose phosphates, where there are 4 isomers all giving off phosphate as a daughter. Can we really separate these out? And this is the best separation shown here of each of the different metabolites that we were able to get, suggesting that we couldn't separate fructose 1-phosphate from fructose 6-phosphate. Now, it turns out phosphate is not the only daughter ion that we're able to get, and we're able to get a unique daughter from glucose 1-phosphate and glucose 6-phosphate, but that's still not helping us out with fructose 1 and fructose 6. And so we had to turn to chromatography to resolve these. And fortunately, fructose 6-phosphate was resolved from the rest. So now we're able to get a very nice separation of these four metabolites by the combination of DMS separation, MRM transitions, and chromatography. And then one last group that we're able to separate, and we've so far been able to separate all the potential or real isobars within glycolysis, but this is lactate and pyruvate, a nice separation. Why I showed this is because this is something where we've done real application in vivo, where we're using DMS to measure metabolic flux in mice. And for these experiments, nude mice were implanted with tumors and allowed to recover in the tumors to grow, engraft and grow. Then they were infused with uniformly labeled 13C glucose under basal conditions. So all 6 carbons of the glucose had 13C in them. The mice were then sacrificed, and tumor, muscle, and lung were freeze clamped and isolated. The plasma glucose was analyzed for the enrichment, and we did prove to ourselves that we were in the steady state. Tissues were ground under liquid nitrogen and extracted with cold 50% methanol. The insoluble debris was removed by centrifugation, and then we analyzed them by LC-DMS-MS mass spectroscopy. And to do this, we used the C18 column with isocratic 40% acetonitrile. So you can imagine that we're really not going to get any significant separation at all. We just did this as kind of a best case scenario to see, we just wanted to spread the peak out a little bit and see how good our method was. Isopropanol was used as the DMS modifier, and we just measured the parent-to-parent transitions for lactate and pyruvate. And just some animal-specific information. These are 3 months old, all the same weight. The tumors weighed about 220 milligrams. Our plasma enrichment was only 5.6%, and the glucose concentration was 110 milligrams per deciliter, which was the same as what we had started with. So we're not perturbing the system, we're just looking at basal metabolism in these different tissues. And when we looked at tumor from a control animal, and we looked at our lactate separation, we were delighted to find that there was virtually no contribution from pyruvate to this, despite the fact that we have no column separation. It was 0.038% contribution, so that's something we can certainly work with. And shown here is the lactate spectra for the samples with and without label, and you notice a completely flat M+3 peak without label, and then a very nice M+3 peak with the label. And when we looked at the pyruvate enrichment in lung, muscle, and tumor, they were all about the same at about 2% enrichment. But when we compared lung and muscle to tumor, muscle and tumor had a much higher enriched lactate concentration. Because we know the precursor pool, that is the plasma enrichment of glucose, we can measure the relative flux of glucose from into glycolysis into these metabolites. And again, there wasn't that much difference between lung, muscle, and tumor, but we saw actually much higher percents of enrichment in the lactate, up as high as 80% in muscle and tumor, suggesting that there was some non-mixing of the pyruvate and lactate pools. So, in conclusion, we think that careful analysis of metabolic flux may reveal many metabolic insights relevant to both normal and disease states. And doing mass labeling experiments is really key to sorting some of these things out. DMS is a very powerful and flexible tool for quantitative analysis of many challenging isobaric species. Orthogonal DMS ion prefiltration may greatly expand metabolomic analysis fidelity, while at the same time decreasing run times. And the addition of DMS to mass labeling strategy extends the number of ordinates for metabolic flux analysis and greatly expands its practical range for in vivo analyses. And oops, see if I can go to the last slide. With that, I'd like to acknowledge in particular Orlando Yarborough from my lab, who's done a lot of the work alongside me trying to learn how to use DMS to achieve these separations, and we are greatly aided by Anthony Romanelli and Keith Goodman from AB SCIEX. And with that, I will pass it over to Jillian and be happy to answer any questions.
All right. Thank you, Dr. Kibbe. So our first question is for you, actually. What drawbacks, if any, do you see for adding DMS to a method?
Drawbacks. So I think once it's already developed, there aren't any drawbacks, with the exception of potential loss of signal. There is a little bit of reduction in signal intensity occasionally, and we actually see increases occasionally. So it turns out to be a pretty good way of doing things as long as you're not limited by signal. Again, a couple-fold reduction in signal intensity is not an issue for any of the things that we're looking at, and so it hasn't turned out to be a problem for us. Another potential limitation is right now the technology does take about 20 to 25 milliseconds dwell time, or dwell plus delay time per transition. So you may not be able to get as many transitions into a peak if you're really working at high number of MRM transitions, but that's all something that can be worked with.
Okay, and second question, is DMS powerful enough for separating compounds to completely replace liquid chromatography?
I think in many cases it might be able to. Now, I did show the example of glucose, fructose 1 and 6-phosphate, where we couldn't completely resolve them just by DMS or MRM alone. And I think there really is a role for liquid chromatography. I just would like for a much simpler, high throughput role, and so reshaping what LC is doing really changes how you think about it and how you progress, and it makes it much easier just to solve a couple chromatographic problems rather than trying to separate everything from each other.
Okay, next question. How easy is it really to find a separation voltage compensation voltage pair for different compounds?
Well, that question really depends on what you're trying to do. The technology is very easy to use. Just doing an infusion, you can easily find an SV-CoV combination. Finding an optimal combination where you're separating your compound of interest either from background signal or from a potential isobar, that can be challenging, especially as the intensity may drop off. You can't just necessarily choose a single separation voltage and expect to use that for all your metabolites, or at least the ones that we've looked at. You're more likely going to have to scan out the full SV-CoV range to get a sense of what's going to work best in your situation. Now that said, the SV-CoV combination is not set in stone. It's not like an MRM where it's completely consistent from machine to machine. The front-end conditions will affect your SV-CoV, so your flow rate, your organic percent, temperature during ionization can all influence the DMS separation. So for each method, you will have to retune an individual SV-CoV combination.
Okay, and this next question might be one for Dr. Aiello. How many transitions can you practically monitor while running using the DMS?
Yeah, so it really depends on the peak widths that you have, the chromatographic peak widths that you have. What you want to shoot for is roughly 10 to 15 points per peak using the 25 millisecond dwell time. If you have a lot of analytes, it's possible to use scheduled MRM to schedule that that acquisition, and so your duty cycle can really be optimized. So for longer runs with a lot of analytes, you can get up into the hundreds of analytes pretty easily. It's a little difficult to answer without really having specific conditions, but it can go to very high numbers if you're doing scheduled MRM.
Okay, and one last question, because we are a little after, well, noon Eastern, and we're almost out of time. If I have an AB SCIEX 5500 series MS, can I upgrade it with Selexion technology?
Yes, so if you currently have a 5500 in your lab, either a triple quad or a Q-trap, that can be upgraded with Selexion technology, or obviously if you're buying a new instrument, it will be, we can include it with the new 5500 as well.
All right, that is all the time we have for today. I would like to thank both Dr. Aiello and Dr. Kibbe for their presentations. Thank you to our participants for being a great audience today. I'd like to remind everyone that each webinar does get archived at CNN Online and the sponsor's website after the live webcast. So those who could not join today or anyone who had technical difficulties can access the information and discussion when those go up. Be sure to check CNN or CNN Online for information on the next edition of CNN webinars. Thank you On24 for technology and production services, and thank you AB SCIEX for the sponsorship that made this interactive webcast possible. For CNN webinars, I'm Jillian Kemsley. Have a nice day.